like Angiotech and Boston Scientific, they licensed this technology, but put a different anti-cancer drug in this case Taxol, though there's others you could put in. And there's a problem in heart disease that you may know about. Probably one of the most common ways of treating heart disease is a stent. It looks like a Chinese finger puzzle. But if you put it in, something like 60% of the time, 50% of the time, you get a problem called restenosis, smooth muscle cells proliferate. You can stop that if you gave an anti-proliferative drug like Taxol. But if you give it systemically, the amounts you need are enormous. It would probably kill you. So what they now do, and this is used in millions of patients, they just coat these stents with a polymer and, say, Taxol, and these are called drug-eluting stents, and they're used all over the world. I want to give you a second example, again, about not necessarily taking things from your clothes, from your wife's clothes or whatever, for a medical implant. And that relates back to what I talked about in the very beginning, where we would make microcapsules or nanocapsules, as Maria pointed out. And that is, if you make things really small, there's a couple of problems you run into. One is that macrophages can start to eat them. And two is they can stick together. So what we did, and a few other people did, this is from a paper we cited, that's been cited in Science we wrote with Vladimir Torchland. It's been cited, I think, 3,600 times, is to add polyethylene glycol, which is a substance that's very hydrophilic and maybe helps give you a disguise. And we showed in this paper that there are nanoparticles. Again, some of this is old. This was a 1994 paper. That's atomic force microscopy. This is quasi-elastic light scattering. But what's very exciting to us is that you could take an orange dye, put it in these nanoparticles, and use different chemistries and different lengths of PEG, or even using a citric acid binding approach, you could have a triple chain. But what you want to notice is on the left, top upper left, if you don't have PEG, even after an hour, look at all the little particles that are eaten. Whereas on the right, the other three, there's either few or none. And so we thought you could use this approach to protect these nanoparticles further. And only Faragut's when he came to our lab, even would go further and put targeting molecules on. You know, I always worry that I don't explain this very well. But I was lucky a number of years ago that Nova, a TV show in the US, they came to our lab and did a show called Making Things Smaller. And they made a video that I asked them to give me, and I will just show it to you, because they explain it far better than me.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That, in turn, gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
I should tell you that a lot of the clinicians I work with tell me it doesn't blow the cell up quite like that. But the idea is you could use this, and people are now using it for small molecules. And of course, what's exciting is that you might be able to use it for genetic medicines. In fact, many people, like Inder Verma, when he was president of the Gene Therapy Society, and Phil Sharp, my colleague at MIT, who's a Nobel laureate, have said that this is the biggest problem in gene therapy today. So if you look at, and I'm going back now to the late 1990s when we did some of the work I'm going to mention now, but you know, people have used viral vectors. They're effective, but they, as you may know, have run into some safety problems. And then non-viral materials like polymers or lipids, which have been less effective, but are safer. And so what we wanted to do is to see if we could help on this. Now, I wish then and even now we understood things well enough that we could do a totally rational design, and maybe more is known, and maybe artificial intelligence could be used. But going back then to the 90s, you know, we thought, well, another way of doing this is if we can't figure it out exactly, maybe we can figure out ways to synthesize very, very large numbers of materials that might have desirable properties. The problem is when we started doing this with polymers, you probably all know this, that there's many steps that it takes to make a polymer, because you have to take a monomer, protect a functional group, deprotect it, purify, and so forth. So it takes a long time. So Bob Grubbs, who won a Nobel Prize, was a friend of mine. He was at Caltech. He said, Bob, my very best postdoc, Dave Lynn, would like to work with you. And Dave actually came, and we thought, well, maybe what we can do is come up with single-step methods for synthesizing large numbers of materials that had cationic or other kinds of desirable properties. So what we would do is just do this conjugate addition of amines to diacrylates. But the nice thing that David came up with was these all involved commercially available starting materials. You can do the polymerization in a single step, and there were no byproducts, no protection or deprotection. So for the first time, you could make a lot of these in very high throughput. And he and Dan Anderson did just that. We actually took, we had robot systems. We took 94 amino, I won't go through each of these, 94 amino monomers and 25 diacrylate monomers, and made over 2,000 polymers. And then we also came up with high throughput ways of using cell lines with the firefly luciferase as a marker, and showed that we could actually get, one of the references that people use then was polyethylenamine as a synthetic material, and these, we had found 46 that worked as well as that. And even a better one than polyethylenamine was what was called lipofectamine. And here you see us doing studies in human embryonic stem cells, and this is one of them, and it worked very well. And this would lead to many different reagents. These are just a few that are now used. Now, of course, the other thing that you could think about and that we thought about were lipid nanoparticles. Our lab had worked on liposomes or lipid nanoparticles, actually originally for vaccines going back almost 40 years. And if you look at, this is today's version of what you might think a lipid nanoparticle would be, but basically you have cholesterol, you have the structural lipid, you have the PEG lipid, that's based on some of the things I mentioned earlier. And finally, you have an ionizable, what's called cationic lipid. Just a little bit of background. So cationic substances in the area of polymers, Wu and Wu actually did this originally in 1987 using polylysine. But the problem is, even though what happens is that if you have a positively charged polymer or lipid, that's good in that it can complex the nucleic acid, but sometimes that causes toxicity, and there are other issues as well. So what Dan Pack, who came to my lab from Francis Arnold's lab, did is, working with Dave Putnam, is we created ionizable polymers. And the whole idea is these would be positively charged at acidic pH, so they could bind the nucleic acid, but they'd be neutral at blood pH. See, if something's positively charged at blood pH, it can cause what's called complement activation or other bad effects. Also, this type of charge effect helps endosomal escape. So this is the idea of an ionizable substance. And so we did it with polymers, and then Peter Collis's group did it with, that Semple et al. Did it with lipids. At any rate, going back in time, I mean, and we did these about simultaneously, Dan Anderson in our lab said, well, look, if we can make all these polymers, could we make lots and lots of different lipids? Again, if you go back to 2000, there were probably only 40 different lipids that had ever been used in liposomes. So Dan asked, could we make thousands? And again, using what Dave Lynn did, that's exactly what we did. We would go back and use the same chemistry. And here again, you could have large variation in R groups. You can have variable tail lengths, a number of tails. And what we did is we collaborated, one of my students, I was also asked to be on the founding scientific advisory board of Alnylam, and one of my students, Kenneth King, led a lot of these efforts. And what we were able to show is that with specific siRNAs, you can actually tremendously knock down LDL cholesterol. This is an animal study we published in Nature Biotech. But you can get, with the right one, close to 50% reduction. So you can imagine if you had a 300 mg cholesterol, now you're down to 150. At any rate, we kept wanting to make these better and better. So we created improved lipid libraries by taking some of the best amines from the original one, and then we would use an epoxide chemistry to generate new libraries. And here what you see is when we do that, is that if you go to LNP01, that's the original one we used to knock down the cholesterol, you can get knocked down, say, with 1.5 mg per kg. But with the newer one, you can actually go down to 0.003 mg per kg. The reason you want to lower the doses is because you'll get off-target effects. The lower you can do, the better off you'll be in terms of safety and especially if you can keep the efficacy the same. So what happened is, over a multi-year period, our lab, actually a company called Acuitas and Alnylam, would keep synthesizing these different things and keep lowering and lowering how effective you could make these. And as a result of all this work by all of us, Alnylam, again led by Akin, who I mentioned, my student, came up with what Maria said, which is a treatment for ATTR amyloidosis. This is humans, and this just shows you the enormous knockdown, and it was quite safe in humans, and that you could get silencing, and this got approved. It's called Onpattro in 2018. Now again, I'll go back in time, 2010. Another thing I started thinking about was there may be other things that you could use these things for. And I was visited by a chemist, a biologist at Harvard named Derek Rossi. And he had done studies where he took RNA, modified it, and were able to change the nature of certain regular cells so they'd be like what are called iPS cells, like a stem-like cell. And he was interested in starting a company and came to see me. I'm not sure necessarily why, maybe because of delivery. But anyhow, we decided we'd start a company. I got some of my students involved and former students. And that company is, as Maria mentioned, Moderna. And the whole idea here is if you take protein therapeutics, they had become very successful, but it takes a long time to make them. Like take eggs, for example, that you grow up to make flu vaccine. The problem with that is you have to guess. So you have to start a year in advance to grow them up. And of course you may guess wrong, like H1N1, H1N2. With this, if you make messenger RNA, put it in a nanoparticle to protect it, inject it in the body will do all the work. It will make the protein. And not only that, you might be able to treat diseases that were never treated before, like ones that are intracellular or membrane-bound proteins. So we started Moderna, and I suppose just like me, people kept criticizing Moderna, which was sad to me. You know, the newspapers would criticize it, scientists would criticize it, and stock analysts really ridiculed it, said the company was way, way overvalued. This is, you know, the first 9 years. Then, of course, COVID hit, as you all know. I just want to show you what the Moderna scientists did. Some, like I said, who were my students. January 11th, the Chinese scientists published the genetic sequence. It took less than 2 days using this technology to finalize the messenger RNA design. A little over a month more to put it in the nanoparticles, ship the vaccines to NIH for testing, and the first patients were treated in mid-March. You know, when you do clinical trials, the first thing you do is what's called a phase 1 trial. That's a small population of patients for safety, to find the highest safe dose. So we had the data on May 19th, 2020, and you're a public company, you have to announce it. And I thought the data looked pretty good. You know, we had antibodies, it looked safe. So I thought now finally the media would, and the analysts and everybody would be happy. Well, let me show you the front page headlines of our local newspaper, the Boston Globe, that day. This is not how you do science, and you can see these pictures on the front page. So it was kind of disappointing, but I want to go 6 months after that. After you do a phase 1 trial, then you do what's called a phase 2 trial, and then a phase 3 trial. The phase 3 trial was 30,000 patients, 15,000 placebo, 15,000 treated. It's double blind, you don't know who gets what. So in mid-November, we had the data on that. We didn't know, but I'm on the board of Moderna, we broke the code. And it was actually it's still one of the most exciting moments for me as a scientist in my life. They basically, when they broke the code, it shows that of the 15,000 patients that were treated, not a single one went to the hospital. Not a single one died, and it was 94% effective. And now this is used all over the world. And if you look at the data, it's been pretty remarkable. This is the US from the CDC. You see the difference between unvaccinated and vaccinated. And if you look at effectiveness, what you also see is that Moderna is 94% effective. Pfizer and BioNTech, that's also made using mRNA and nanoparticles. That's 91% effective. Those are the only two that are FDA approved out of over 50 that people have tried. They've also had over 209 different treatments for COVID. Only one of those 209 have been approved. So it's not been an easy thing to do. And I like to think this is just the tip of the iceberg. One of the things that Moderna and Merck recently announced is maybe you can create what's called personalized cancer vaccines. So here we took 157 patients with advanced melanoma, and you do PCV, we call that personalized cancer vaccine. And that means you look at the patient's individual cancer, incorporates a genetic code of what we call the neoepitopes, that's what makes them abnormal, into an mRNA vaccine, exactly the way it was done with the COVID vaccine, mRNA and nanoparticles. And you do, again, a double-blind clinical trial. Half got Keytruda, which is Merck's actually immunotherapy drug that's been incredibly successful, and half get Keytruda plus PCV. And the data were really remarkable. The risk of recurrence or death was reduced by 44%, highly statistically significant. I thought I'd spend the last few minutes of this going over some future things. So in 2012, way before any of this stuff happened, I got a visit by a software entrepreneur. But he and his wife have this foundation, and they want to do good things for the developing world. Here's a picture of me and the software entrepreneur, which, if you don't recognize, is Bill Gates. And he said, we have a whole bunch of projects with him. Almost maybe half the lab is doing that now to try to help people with different afflictions in the developing world. But one of the things that Bill asked, he said, people in the developing world don't come back for their second injection, almost never. Of course, he didn't know what would happen in the United States later on, but at any rate, so he said, well, could we come up with a way to create what we call self-boosting vaccines? And I had thought about this before, and the idea, if you look at this red dot, and I've been trying not to point with these, but if you look at the red dot, that could be the vaccine. Maybe we'll cover it with like a tiny eggshell almost, a rectangular eggshell. But what we could do by changing the composition of this outer part of the eggshell, we could make it break at different times, like time 0, well, that's easy, but 1 month, 2 months, whatever you want. So how could you do that? Well, what we did is we took, again, FDA-approved material, lactic glycolic acid, but the beauty of it is by changing the molecular weight of it and changing the copolymer composition, we can make it break at almost any length of time. I'll show you that. We came up with a sort of a picoscale fabrication method to make bases, to fill it with a pico filling system and seal it. And this was published in Science, and there have been 5 other papers since then advancing this in different ways. But just to show you some data, here is one animal, but we're giving the animals cocktails of ones that degrade it or break at different times. And you can see by changing the compositions, each line represents a different one of these. But there's 11, it's one injection, but there's really like 11 boosts. So that's one of the things that maybe will happen someday. Also, we've collaborated with Mark Krausnitz, one of my graduate students, to put these in microneedles. I don't know if people have seen microneedle patches, but we've done a lot also in transdermal delivery. But microneedles don't go in very deep. They go in just deep enough to deliver something, but not so far that they cause hardly any pain. So what we've been doing is putting these in, and then this is like a little, if you look on the left, it's like a little Band-Aid, and you put it in, and our hope is someday these could be shipped all over the world to help people on a vaccine. And to that end, also, this happened before, we got money from BARDA in the United States, a government agency, and they said, could you ever make microneedle, and again, this was more for, with Bill, the idea was polio vaccines originally, but we've done mRNA vaccines now too. With BARDA, they were also interested, what if there's a new Ebola outbreak or something else? Could we have a way that we could print, like 3D print, microneedle systems, in remote areas that are hard to get access to? So we just published 3 weeks ago in Nature Biotech a way to do this. I'm not going to go into the detail too much, but here you have a mobile 3D printer. It's very tiny, but it can be shipped all over the world, and you can make a vaccine ink, which we did in this case, of mRNA polymers. We found the right polymers to help stabilize this, polyvinyl alcohol and polyvinyl pyrrolidone. And you could make patches just like I showed you on the last slide. But what was exciting in that paper is that one of the other big issues is shelf life. So these vaccines appear, from what we've done so far, to be stable at room temperature for 6 months and 37 degrees for at least 1 month. And this data just shows the microneedle patches on your right compared to intramuscular injection, which is the gold standard. Again, this is in animals on the left. I thought I'd end this talk by telling you about one other sort of futuristic thing we've done, and you have people here doing great work in this, and that's tissue engineering. And to motivate that, here's a little baby who's dying of liver failure. My colleague, Jay Vicanti, and I had this idea, probably close to 40 years ago, that you can make new tissues and organs from scratch by combining materials like biodegradable scaffolds with pretty much any cell. This is from a paper in Science. I think it's been cited about 9,000 times. But the idea is with the right scaffold, the right reactor conditions, you could actually make pretty much any cell type. And again, we synthesize new materials like new polymers. We could use, in some cases, FDA-approved polymers like polylactic glycolic acid, but we also, Denise Barrera in our lab, also synthesize new ones because sometimes you need the cells to sit on. They won't attach unless you have certain cell sequences like RGD sequences and so forth. So we also made materials with an NH2 group by adding lysine to them, and then through, say, a carbodiamide reaction, you can attach whatever you want. And then you make them into fibers like this. And we've actually worked with companies to treat patients. And I thought I'd just show you one example of that. This is a little baby who's very badly burned. Here's the product, which are neonatal skin fibroblasts. On this, it can be cryopreserved. We can put it in the child at the time of injury, that's time 0. Here he is 3 weeks later. Here he is 6 months later. And this is now approved by the FDA for patients that are burned or have skin ulcers. Not only that, the other big area, of course, to me, when we did this, is that not only could you make new tissues or organs, but maybe you could make organs or tissues on a chip or in vitro systems. So you could someday do high-throughput drug testing, but also reduce the amount of testing someday in animals and maybe someday in people. To that end, even over 20 years ago, Gordana Vanyak and Lisa Fried in our laboratory took heart muscle cells and put them on a scaffold, and they were able to get this characteristic structure. And you can even take a video of the heart. And this is in vitro, but it's actually 6 million cells. And we're not shaking this or anything, but 6 million cells and it's beating like a regular heart. And you can actually take an EKG of it, and it's like a regular EKG. And Gordana and her colleagues actually started a company to do drug testing on this that's working with quite a few other companies. And I won't go through the last slide, but I just wanted to put it up just to show you where all this is going. This is like all the different engineered tissues that we and our lab members and former lab, or not all, but some of them, I think 10, either organs or tissues on a chip, even working on brains on a chip with Liwei Sai, but all kinds of different ones that are at various stages of either animal testing, human testing, or even trying to create organs and tissues on a chip. Again, a lot of this is all kinds of work. I hope lots of people, and I know people here, like in Dr. Hilburn's group and others, are doing work on that. So I just, in closing, want to say what a pleasure and honor it is to be back here. And I really appreciate people listening to hearing my struggles, but also my dreams of how one can develop new chemical principles and new engineering principles to relieve suffering, to prolong life, and hopefully to make the world a better place. Thank you so much for having me.
Thank you, Prof. And for me, you really show when you bring science and engineering together, just like the Faculty of Science and Technology, what we can be able to do when we're joining forces. You have done that in your own laboratory. I think we can do something similar within our faculty. Now, open for questions. Jöns.
So I like your history story and the beginning there of trying to pursue this patent of macromolecules delivered. But you published the papers in 1977 and you get a patent approved 82 or 83 after publication. What did you keep any secrets to the patent, or how did you do that?
Well, that's a very interesting question. Let me go through that. So, no, we filed it. We filed the original one before we published the paper. But, you know, the patent, and I'll tell you an interesting story in a second, which relates even to things that you're doing. So that patent, you know, the examiner kept rejecting it and rejecting it. So you can file, so the key thing on the patent is the date you file it, not the date you get it. So we got, like I say, 5 or 6 years of rejection. By the way, the original tissue engineering patent, that got over 10 years of rejection. But actually, that was a good thing, because this is an interesting thing. You know, they changed the laws. So it turned out that because that it got rejected, you know, that the date, it used to be, well, now the rule is 20 years or 21 years from when it's filed. But before 1995, it was 17 years from the date of issue. So it was actually ended up being useful, because some of the products that came out, tissue engineering take a long time, as you know, to take off. So finally, you know, and so it actually covered some of the things that people did, like artificial skin. But very good question.
Kalle?
Thank you very much for a very interesting presentation. So the mRNA vaccines is truly a spectacular breakthrough. So do you think this will be the dominant way of making vaccines in the future? And if so, is there a solution to the fact that these vaccines need to be kept in the cold, which is a disadvantage compared to the traditional vaccines?
Yeah. Well, of course, I'm highly prejudiced, so on the question. But I do think that it's certainly going to be one of the key ways to make them. And certainly if there's an emergency, it's got to be the way to do it, because none of the others can be done nearly as quickly as we saw. And of course now there's better ways, you know, and now I hope we'd at least be prepared. And you know, but the beauty of this, as you can see, is you can literally design it in 2 days, and the manufacturing techniques compared to others are much simpler. And like I say, someday I hope we'll even be able to have these patches and other things. Your second question is a very good question, and I probably should have said, and I believe this, I hope part of what I said today motivates others, and I know people are already motivated, to do much, much better than we've done already. And you certainly raised one of the issues, which is shelf life and stability. And there's lots of other challenges, like targeting and other things as well. Drug targeting to specific cells and so forth, but crossing the blood-brain barrier, which some people are working on, and so forth. But I would say the answer to your question is yes. I mean, already Moderna has more shelf-stable ones. I wouldn't be surprised if BioNTech, that's the other company that's been doing a lot, has too. And as we can see from even what we published in Nature Biotech just a couple weeks ago, when you make them in a certain form with certain polymers, we can, you know, greatly enhance the shelf life. So I expect that will be a solved problem. But there's other problems that may be even harder to solve, and I think good chemistry and good engineering will contribute to that. Like you're hearing a prejudiced view.
Thank you, Professor, for your talk. Very interesting. So now you've been presenting several projects where people have been skeptical about previously, but now are helping many millions of people. Which projects are people skeptical about right now, but which you think would be helping millions of people in the future?
Well, I mean, that's a very interesting question, and I think you get skepticism all the time. I think some of these things, you know, that you could recreate organs or tissues on a chip, I mean, and people might be right to be a little skeptical. You know, we're actually, like I said, even trying to create brains on a chip. And I think stuff like that, you know, you know, I could certainly see skepticism, gastrointestinal tract on a chip. But, you know, so I think my experience, especially at the convergence of, say, chemistry and engineering on the one hand and medicine on the other, is that you do get a lot of skepticism. I mean, I just think that's human nature. So I think this is certainly one area, but there are certainly going to be lots of, anytime you do something new, you know, maybe I think it invites skepticism. And I think that's just what I've seen over and over again.
One question over here.
Thank you, Professor Lager, for
the talk. It's very interesting and very inspiring. My question is, in the drug delivery, like the siRNA, we know there are like another technology like exosome. So, in your opinion, like what is the position between the exosome and the LNPs? Which is better, and yeah, what do you think about it? Thank you.
Yeah, so the question for people that aren't following this, you know, so lots of people are using different sort of nanoparticle delivery systems, you know, lipids, polymers, and others too. One of those is exosomes, and people are putting drugs in exosomes. I think it's an interesting area. I think, actually, I thought what would be interesting if somebody could do it, and we're not, but it's like almost a synthetic exosome to try to understand what it is about that has interesting properties. I also think some of the interesting things about it, I don't think you'll ever see a one-size-fits-all, you know, of one material that will be useful for everything. But some of the things that have been interesting to me, and I'm hardly any expert, you know, are exosomes getting across certain barriers, like there's been some reports about possibly crossing the blood-brain barrier. And I know that's something that's being worked on here too. But I think that there are, you know, there are probably going to be places where exosomes, as they're further studied, will be useful. And I think that's probably a good thing.
Maybe one more question?
Can I ask a second one?
I'll try mine first. But we have all these skepticism of starting projects, and it worked well. Could you give an example of a project where you had a lot of people really believing in it, but it didn't work so well at the end?
Yeah, sure. Actually, I mean, maybe I'd have to think about all the ones that are maybe like that. But I also had this idea, I'll just give you an example. I also had this idea once that, you know, because you've seen me do a lot on delivering molecules, macromolecules, and so forth. So I had this idea, actually also a long time ago, that maybe we should figure out ways to get rid of molecules that are bad, not just deliver ones that are good. So one of the molecules I had in mind was, and one of the things that's been helpful to me is I get all these great post-fellows coming from the hospitals. One was a fellow named Art Levin, and he was a neonatologist. And he said, well, what's really terrible is he sees all these babies that are jaundiced. They have high levels of bilirubin. So we'd already been doing some work with different enzymes to remove heparin or other things. And I talked to Alex Slivanoff, who's one of my friends, and who's an enzymologist. And we thought, well, I said, you know, there's these bilirubin enzymes, bilirubin oxidase. Maybe we could make a little sort of reactor to get rid of the bilirubin in the thing. And we did that. We actually published it in Science, and we showed that you could use this approach to get rid of bilirubin in jaundiced rats. So, you know, that looked all very good, but, you know, around the same time, some people came up with another way of getting rid of bilirubin, which is using lights. Which process was photoisomerization. And I have to admit, when I saw that, I thought that's probably better than our approach. So, and that is actually the standard of care today. If a baby has bilirubin, they put them, or jaundice, they put them under lights. So even though we published it in Science, and I think it got a lot of interest, you know, it didn't lead to anything. But that's part of, I mean, you know, that's part of science. But that's an example. That's an interesting question. I don't think I've ever gotten it.
Well, I heard similar from Professor Rogers. Sometimes we just over-engineer things.
Yes, that's true. But I mean, I haven't never occurred to me to use lights. I wish it had.
Just the last question for you.
Yes, you made these pegylated particles, and I know that a lot of people have antibodies towards PEG. How does that influence the drugs, pegylated drugs and pegylated particles? I've heard about it, but I cannot understand if it has any effect or not. The PEGs, they come from shampoo and skin creams and everything that creates the antibodies today.
Well, I don't know that there's a really good answer to that. You know, I can give you my answer as a layperson and maybe add a little bit to it. You know, I don't think overall it's had a major effect, but, you know, when you look at every so often for different people, there are these rare effects, right? That maybe they do get some reaction whether it's to that or to the lipid or whatever. So there have been certainly cases where people have had side effects. I should have probably pointed out, because it's important, it's not like the mRNA vaccines have no side effects. But if you compare the side effects, and this is in clinical papers, that you get from mRNA COVID vaccines to what you get from flu vaccine, you actually get more side effects with flu vaccine. And of course, they don't have PEG. But I don't know I don't think anybody really knows what the culprit is. But your point is right. So, one of my former visiting scientists, Xiaoli Zhang, who's at Cornell, he and I are working on PEG substitutes, like, which are squid ionic polymers. And they, at least in animals, get absolutely no reaction. So maybe someday something like that, will make a difference. So, but it's an interesting question. There's just so much we don't know, and especially when you get into human beings, it gets more and more complicated.
So with that, let's thank Bob once again.